Hi, everybody. We're back again with another edition of What's Up with FOXG1 Research. And we have a really exciting announcement. We have a new research project to let everybody know about. We're starting to do experiments, moving into translational science. This project is about CRISPR A. And we are here with the Fink Lab to tell us all about how they're using CRISPR A for FOXG1 syndrome. So welcome Kyle Fink and Peter Deng, and let's begin. So tell us a little bit, well, first of all, you're from the Mind Institute at UC Davis. So tell us a little bit about yourselves.
Yeah, so thank you again for having us. And like you said, we are very excited to start our research in FOXG1. So my name's Kyle Fink. I'm an assistant professor in our Department of Neurology. And I have joint appointments in the Mind Institute, in our Institute for Regenerative Cures, and our stem cell program, and our gene therapy center. And really what my lab focuses on are rare genetically linked neurological disorders. And neurodevelopmental disorders is really one of the areas that we focus on. And the main thing that we try to do in the lab is really work on the translational front, or using kind of the leading edge technology to develop therapies. And the type of therapies that we want to develop are really targeting the underlying genetic conditions. And so FOXG1 is the example, or an ideal gene that we're interested in working on, because we think that we'll be able to really go after the endogenous expression of this gene to induce a therapeutic effect. I would like to introduce an extremely talented postdoc, Peter Deng, who will be really leading this project at the bench, and really working on these novel tools.
Hi, everyone. My name is Peter. As Kyle already said, I'm a postdoc with his lab. A lot of my background has always been with these basic rare genetically linked neurologic disorders. Previously, I worked with Angelman syndrome extensively, with Huntington's disease extensively. That's actually how Kyle and I became involved together. Was we had a Huntington's project using basically these artificial transcription factors, basically these proteins that can bind on the DNA and up and down regulate, which CRISPR A falls underneath that umbrella. So I did a lot of that during my graduate career, and I stuck on for my postdoc to basically continue on to these projects, which is, as I started my postdoc career, FOXG1 was one of the things that basically came forward, and I was very excited to work on this project in that basically how we understand FOXG1, and like Kyle said, it's really an ideal system for a CRISPR activation approach. And we're very excited to basically apply the tools that we've applied to different diseases in the Fink Lab, basically to FOXG1, and incorporate it into our current pipeline.
Well, it's always exciting for us to hear that FOXG1 is an ideal gene to work on. So we're excited to learn more about how you came across FOXG1 and why it is so interesting to you. But why don't you tell us a little bit about, well, why don't you start telling us a little bit more about the Mind Institute?
Yeah, and so my lab we're affiliated with the Mind Institute. And really what we try to do as a group is working with the very talented basic scientists that are at UC Davis, and we have an unbelievable genetics department and our genome center where our close collaborator Dave Siegel works, in terms of understanding genetics and understanding how genes interact with each other. And then we try to kind of bridge that gap all the way to our wonderful clinicians at the Mind Institute. And so for those that don't know, the Mind Institute is a world-renowned center that studies developmental disorders. And so they've had a focuses in many different rare diseases over the decades. And they have some of the best clinicians, some of the best centers that are able to see children, help diagnose them. And really one of their major goals, and where we interact with them a lot more, is understanding this diagnostic odyssey that a lot of patients and families are really going on, where it may take years for people to get an accurate genetic diagnosis of different conditions. And so really what the Mind Institute is doing in their human genetics program at the Mind is trying to limit this diagnostic odyssey, instead of having it be like an average of 7 years before maybe you're able to access sequencing and understand underlying genetics, to making that weeks within being seen. And so we communicate a lot with them. And so we try to kind of have this comprehensive front to back understanding the basic genetics, understanding how we can manipulate DNA, how we can regulate gene expression, all the way through, getting to interact with the patients and the families and how they're being seen at the Mind Institute. And so my lab kind of exists within that spectrum, where we try to do things that have high translational relevance. And so working within the human genome, trying to get cells derived from the patients that we can turn into induced pluripotent stem cells, which I know FOXG1 is well versed in creating cells and understanding and studying cells. But really what we want to do is be able to take those materials and take that information and that basic science and try to push it forward, within the clinical realm.
It's wonderful. And, we have to thank our CFO, Ryan, for really finding your lab and nurturing this relationship to get us to this point. So it's very exciting. Some of our, the viewers of this are donors, parents, and they may not be familiar with the word translational means. Can you explain what is that progress from basic science to translational? Why is that so important?
Yeah, and to reiterate again, I think a lot of the projects that we end up taking on in the lab, come from very passionate families and patient advocacy groups. And so we met Ryan, and I think we were immediately hooked on wanting to do something and wanting to work on FOXG1. So when we say translational research, what we really try to think of is there's gaps along the process of finding the gene to having something that goes into patients in like a clinical trial. And there's a lot of steps along the way, and there's a lot of gaps in knowledge that tend to appear. And so what we try to do is kind of fill in some of those gaps. So if you have something that works in a cell or works in a mouse, is there a way that we can, one, show that it's safe, that it's efficacious, that it's going to do what we want it to do, and appropriately be able to judge kind of the benefits to the risk, and then working with, UC Davis is uniquely situated, and where our lab is in the Institute for Regenerative Cures is uniquely situated, where we actually have a lot of clinical expertise and regulatory expertise to take new biologics into clinical trials. And so the California Institute for Regenerative Medicine, which is one of the funders statewide for us, it's a really big driver in cell and gene therapy for us. They created what were called the alpha stem cell clinics. And these alpha clinics basically put in a network of people that can help recruit patients, that can help do the regulatory process, that can help you do the manufacturing and the scale up. And then actually, one of the most rewarding things that we see in our lab is we have a manufacturing area where we can actually make clinical grade products, and they'll get made, and we have, they call it the STEM mobile. It's a really nice golf cart. And they can actually take products that were developed at the bench that we designed, manufacture it at the clinical grade, and they'll drive it over to the infusion hospital, right? Wow. So they just take a short drive across campus and they're treating patients. And so I think there's 47 clinical trials that are being administered out of UC Davis and out of this alpha stem cell clinic, right? So we want to kind of help fill that gap in these rare developmental disorders, right? In these neurodevelopmental disorders, is how do we apply the knowledge, the institutional knowledge of taking things forward to some of these rare indications, and how do we really push, these interventions that could have a transformative effect.
Yeah, and so Kyle, I know we connected relatively early. Very excited to get FOXG1 in your lab and in UC Davis's peer review, given everything that you guys can do there. So let's talk a little bit about the modality that we're talking about here, CRISPR A, and maybe give some examples of using those techniques in other neurodevelopmental disorders that are similar to FOXG1 that got you excited about this. We've all seen, I know our community has seen the recent publication that you guys submitted and got sent up for Angelman, and that's very exciting. But what can you tell us about CRISPR A and the prospects of FOXG1?
Yeah, and so I'll actually turn this over and let Peter explain a little bit about some of the strengths that we view that CRISPR activation technology may have, and why that's very applicable to these rare developmental disorders.
Yeah.
If you will, Peter, I think a lot of people hear CRISPR, and they don't know the distinction between, CRISPR and CRISPR A.
Yeah.
And so this new project that we're doing together is a CRISPR A project. So if you could sort of let everyone know what the distinction is and the difference.
Yeah. It's actually one of those things that's really important to let people know, because it's actually pretty crazy, I think, with how science has been communicated to the public, because especially with CRISPR, as we know, there's these ideas where we can genetically modify anything now. And I think the popularity of CRISPR coming out has really bled into popular media, where, grandparents are talking to you about CRISPR now, and you're like, oh, wow, this is crazy.
Yeah,
And
and
I think
they're using CRISPR, but what does that mean for us? Yeah.
exactly. And
Yeah.
I think it's like we thought of this as stuff of science fiction years
Right.
ago, but we're here today where people are actually being injected with this stuff in therapeutic applications, right? And so I think when we think about what CRISPR-Cas9 it was basically a system that you could design it to bind onto DNA. And the way it binded onto DNA was you have your Cas9 protein. That's the stuff that basically would initiate an effect. And you would have a paired guide RNA. Basically, that's the thing that's going to target a piece of DNA. And it leads the Cas9 there. And the way the way Cas9 was originally described was that it would bind onto that thing and cause some type of cut. And it's in terms of its applications early on, it was, well, you could cut DNA, you could edit DNA, which has profound impacts in terms of therapeutic development in modeling and everything else. And something that we were very interested in our group over the last 7 odd years was basically, rather than doing an editing approach, which in itself can be very difficult to do, but it's actually fusing and defanging Cas9. So it wasn't actually cutting, but rather you can pair different things to Cas9 that can turn off a gene or turn on a gene, and everything in between, where you can actually, what we call tunable genetics. And so CRISPR A in this sense means that we don't use a Cas9 that cuts, rather we use a Cas9 that is fused to an activating protein, so that when it actually binds onto DNA, think of like the way DNA is often described is you have your gene and you have a promoter, and the promoter is sort of the ignition switch for that gene. And naturally, you have sort of a natural amount of expression that's going to be apparent there, but you can push it into overdrive. And we can try to use basically a CRISPR activation approach as a way to push a gene to express more of itself, which is highly applicable to FOXG1, in that while there may be one non-functional FOXG1 gene, there's still an intact functional FOXG1 gene. And we know that this is a haploinsufficiency where there's not enough FOXG1 being expressed. So with that in mind, we can try to create a Cas9 activating system, a CRISPR A, to bind onto that healthy FOXG1 gene and cause it to express more of itself. And that's the idea, basically, with this idea that there's haploinsufficiency or lack of FOXG1, we try to make it produce more FOXG1.
And a lot of when we say CRISPR A is still a very broad category of these activator domains. And so one of the things that Peter and another postdoc in my lab, as well as everybody in my lab, has really been focused on developing are these ideas of how do we use those different activator domains? And these activators are actually already factors that are found in our body that regulate other genes naturally, right? So we're kind of just hijacking biology a little bit to say, if FOXG1 is 50%, what can we do to bring it to 100%? And what happens if we take it to 300%, right? Is there ceiling effects? So not only can we develop something that we can try to rescue the normal function of FOXG1, but we can also understand other things, right? So if we're doing traditional gene replacement or gene therapies or other types of indications, are there things that we can learn from these, kind of basic / translational science experiments to understand really what the role of FOXG1 is within these cells? And yeah, so Peter's working a lot on that, and this idea of tunable genetics is something that we're very interested in, right? And how do we make these minor modifications to make a gene behave in a therapeutically relevant way?
Wow, it's incredible. So tell us a little bit of what you've seen so far using this method, this CRISPR A method, in other genes and other disease groups.
So we've worked a lot, as was mentioned, and so we work a lot in collaboration with a gene called, or with a disease called Angelman syndrome. And And all of these different genes that we would end up working on have very unique traits for some reason. You know, and they're always present their own interesting challenges that we have to adapt and overcome. And so what we've been doing with the Siegel Lab as well as the Silverman Lab at UC Davis is trying to understand in Angelman syndrome, there's a unique genetic event in where there's a healthy allele that gets turned off. And so we think that we can introduce a therapeutic rescue by turning this gene back on. And so again, using different CRISPR tools of ways in which we can drive the expression or unlock this healthy gene. We also work a lot in other conditions that have been closely linked with FOXG1 in the past, things like MECP2 or Rett syndrome, as well as CDKL5 deficiency disorder. And in those cases, because in females, because they're X-linked disorders, there's a healthy copy of the gene that's epigenetically silenced. And so we can go in and try to do targeted epigenetic changes to basically rescue the gene expression. So in all of these conditions, there's in Angelman syndrome, in Rett syndrome, in CDKL5 deficiency disorder, there's not enough gene present in the cells that it needs to be present in. And so we use these different CRISPR tools, CRISPR activating tools, CRISPR epigenetic editing tools, CRISPR tools that can modify RNA expression. And we just use them in this kind of institutional knowledge of how we best apply those for unique genetic situations. You know, some that we may have to change, you know, the methylation status of a gene, some that we may have to target an imprinted gene to get it to be re-expressed. And something like FOXG1, how do we get, you know, FOXG1 expression to best mimic the healthy condition, right? Without too much or too little, somewhat of this kind of Goldilocks conundrum that we run into with gene expression a lot. And so all of these different indications, you know, we kind of start, you know, working with cells, working with human cells, patient-derived cells, if possible, where they have the mutations that are, you know, seen in the kids. And we turn those into, you know, what we call like a disease in a dish model, where we can differentiate those into neurons, and then we can evaluate how effective they are. And so a lot of the lab is really focused on developing the tools in this way. And then we have a lot of members in the lab because, you know, it's really cool if we fix a cell in a dish. But that doesn't really probably mean a whole lot for many people, right? And so we have a lot of people in the lab, and Peter's led efforts in this, and another postdoc in the lab, Julian Almai, is really leading a lot of efforts in this now too, is how do we move that forward? And so can we put these things into different types of viral vectors? Can we engineer cells to be able to deliver these types of cargos? And then we work with our colleagues and our collaborators, right? So we can go from the cell to an in vivo system, something that models the disease. And then we work with other great collaborators here at UC Davis. We're incredibly lucky that this is the California National Primate Research Center, and we have wonderful colleagues there that we can try to move everything all the way through to what would be like a clinical trial.
Kyle, let's talk a little bit about administration of this. If we're able to achieve upregulation of FOXG1 via activation, a lot of questions that we get are, like an ASO, how often would a parent need to dose their child? Or a gene therapy, is that a one-time administration? Is that a several? What is CRISPR A? If we can activate the healthy copy to mimic the proper expression, is that a one-time done deal, or is that a continual therapeutic? Do you have any
How
insight?
does it work? Like, how do you administer it? Yeah.
Yeah, so there's multiple answers to that question. And it comes down to the question of delivery, and this is delivery is a topic that Peter's focused on for years with us, is how do we best efficiently deliver these things in the safest way. If we do it with a gene therapy, it would be a one-time administration. It would be something that what's been observed with like Zolgensma and SMA is you give AAV once, and recent publications have come out that say that AAV expression will exist in neurons for decades. And so, if we go early and if we can deliver it and it gets to the cells in the correct manner, this is something that may be a one-time administration. If we have to deliver them in something like a nanoparticle, which, nanoparticles are extremely popular now because of the current situation and development of rapid scientific progress. Peter and I have joked together that we were actually doing a lot of nanoparticle work about 4 or 5 years ago, and we said, oh no, I'm not sure this will develop into anything useful, and we moved away from it, which wasn't the good choice, right? And so, if it's a nanoparticle delivery, this may be something that has to be re-administered. One of the very unique things about using these CRISPR tools is around this idea of epigenetic modifications. And when we talk about epigenetic modifications, sometimes epigenetics is kind of referred to as the fifth base, right, of DNA. So ways in which we can modify something without changing the native DNA structure that will have a long-term effect. And, colleagues and other people have shown in different publications that you can induce a change to gene expression by modifying the underlying epigenetics. And that effect may be long lasting, long after like CRISPR and everything else, all the other machinery is gone. And so one of the, the dreams or the where we're kind of setting the goalposts is how do we deliver these things, in a way that's not going to be a burden to the patient population, and that could have a long-term effect. For if we can do that in FOXG1, we're still kind of understanding that, right? And that's the different combinations of activators that we use for understanding if is it going to be a long, durable upregulation of the gene, or is it going to be, something that we put into AAV and then it's there all the time, but it's a one-time administration. So these are all questions that we're excited to try to answer, and we're excited to keep pursuing. But yeah, it's a pretty cool area to be working in and to see the breakthroughs and advances.
Kyle, what do you think about, with CRISPR, the off-target effects obviously are what is kind of stopping mainstream adoption. Do you feel that CRISPR A, by off-target effects, I mean, your child gets cured for their disorder, but then they get a brain tumor. So with CRISPR A, do you see that as a safer route than CRISPR where you're cutting Cas9?
Yeah, and that's an incredibly important point to make. And Peter did touch on, when we think about the distinction between CRISPR A and CRISPR as a nuclease activity, is anytime that you're inducing double-stranded break repair, right? So when you cut the DNA, it has to repair itself and come back together. Most cells don't enjoy doing that, right? Most cells don't want to repair their own DNA. And typically when they're repairing their own DNA, it's due to some type of insult or damage. And like you said, is a lot of times implicated in cancer. And we give a whole talk. I teach a stem cell and gene therapy class, and we have a whole series of slides that we keep adding to that, is the CRISPR bad things? Like, what are the things that can happen within a negative effect, right? And most of those are associated with off target when we're cutting. One of the advantages for doing CRISPR A and regulating gene expression is we can be very selective in the guide RNA, right? And so we don't have to, we can move them around, and we can select a guide RNA that only will bind in one 20 base pair sequence out of the 3.3 billion base pairs we have in the human genome. And we can test that, and we can very closely evaluate that in human systems to make sure that it's very specific. The other thing that we do evaluate and that we plan on evaluating in this proposal is not only when it binds and it changes, so we can show the on-target or target engagement of FOXG1. So we can regulate FOXG1 specifically. Then we'll look at all the other RNAs as well. We'll look at the whole transcriptome, and we'll say, are there meaningful changes to other genes where Cas9 has bound? Because, there could be an issue with, if Cas9 bound in what we would call a gene desert, it may not have an effect on regulating any genes in a negative way. And so, these are basically the safety steps that we'll work through to make sure that, one, we're doing what we want to do where we want to do it. And then we'll look everywhere else too, and try to ensure that Cas9 is not binding anywhere else. And then secondarily, that it's not causing an effect anywhere else. And so those are some of the experiments that Peter's designed and will be undertaking with, again, human cells containing the FOXG1 mutations.
Yeah, I think that basically anytime you're developing a CRISPR technology or CRISPR approach to any type of disease, there are certain best practices that really need to be followed up because, like Kyle said, there's always the potential that these things could bind elsewhere. And even from the design perspective, we do a lot of this through like well-known algorithms that are supposed to be very specific in terms of its design. And we try to use that as a guiding principle for the initial design. But all of these things, there's always context associated with these different guide RNAs. And we have to treat every single system and everything we design as its own independent thing. And so while we do have a pipeline in lab to basically evaluate and construct these things, we have basically these, like I said, good practices or checkpoints that we have to cross when we evaluate these things. Let's say we find guide RNAs that work really, really well and they turn on FOXG1 X-fold and it's fantastic. But there's a reason why we evaluate many of these at the same time, because just because one works really, really well doesn't mean it's necessarily better than one that doesn't work as well, because that one that doesn't work as well may actually be more selective and it's going to be more effective in terms of driving something that's good versus something that can be too good or go elsewhere and do something else that you wouldn't want it to behave. So like Kyle said, we really want to look at basically where these guide RNAs are going to bind and how they influence FOXG1, but also beyond that, like what are the downstream effects that can occur from it. And all that's really based on using cells that are going to be relevant. So many people will study these in a cell type that's not really relevant to a neurologic disorder. And something that we really want to study this is in, patient-derived cell lines that don't have FOXG1 and actually study how it's actually going to behave in that context versus like an ideal context.
That's interesting. I'm sorry, go ahead, Ryan.
No, go ahead.
Okay, I just wanted to break down the general project milestones and what you expect to achieve. So just sort of laying out how you see this project unfolding and the best case scenario.
Yeah, and so, I think we had a lot of calls early on with Ryan when we first were able to meet him and coming together, and we're very enthusiastic about the potential of studying FOXG1 and where we can take it to. And so this is a one-year project, and we're kind of considering it, a proof of concept project, right? Can we modify FOXG1? Can we regulate FOXG1 in a therapeutically relevant way? Can we develop the tools that would be able to move forward with a therapeutic? And so we've kind of set, different benchmarks and milestones within this. One is, what Peter talked about, can we screen guide RNAs and what we call activators or effector domains that we can get this tunable expression of FOXG1? So we can see, can we take it from 50% to 100% or 150% or beyond to really understand, how do we best regulate the genes with certain combinations of guides? And we're going to do that in, kind of these cells that we use for pipeline evaluation. And then we're going to do it within what we call an allele series of FOXG1 patient mutations. And so where we have a healthy iPSC line, and then we have multiple variants that are pathogenic of FOXG1. And we can differentiate those cells into neuronal stem cells as well as neurons and understand a little bit about what are the differences between the disease cells and the healthy cells and what are those meaningful targets, right? So the easy one is the healthy cell should have a lot of FOXG1, the cell with the variant should have half of that expression of FOXG1. And so the question is, one, can we just go from 50% to 100% in a neuronal stem cell or a neuron that contains a FOXG1 mutation? And then we're going to be doing a lot of other things, kind of within our center where we call it like neuronal profiling. And so, when we look at the healthy cells and we look at the cells with the variant in them, what are the differences? How do they grow? How do they differentiate? How do they talk to each other? And then doing these things like whole transcriptomically, like FOXG1 as a transcription factor, and just as a gene is incredibly interesting for all of the different interacting partners that it has, to understand if there's less FOXG1, what are all of the different networks that may be impacted? And so, a second one is, so the first kind of goal is, can we modify FOXG1 expression? The second goal is, what are the differences between the healthy cells and the disease cells? And then, the major goal for us at the end of the first year is, can we change those differences between the healthy cells or between the variant cells and make them look like a healthy cell? And I think, the ideal goal at the end of this is we would have something that we would consider a therapeutic candidate And then we would want to do stuff, and we would work with the FOXG1 Foundation to understand what's the best path to take this forward. Is it going into an animal model? Is it doing more studies within cells? You know, and you know, tracking that or putting that path forward to really make it look like a therapy.
Yeah, so I want to bring this back to so the science talk is fascinating. I'm sure everyone's really going to enjoy that. And I want to bring it back to the human element of this, right? When we began talking, you spent countless hours along with your peers with me on Zoom. You know, I met in person your lab and Jill. You met my son. And we were attracted to your group for your skill set, you know, your brilliance and the infrastructure that the Mind Institute has to conduct this type of research and move it forward. But what really came through at the end was your general want and need to move these things forward from a therapeutic perspective and help families. We encounter academic institutions often that want to conduct research and get a publication and learn, and that's kind of the end of the road. You guys strike me as people that want to make a difference. So can you talk a little bit just about the culture around that in your lab, in your peers' labs, of really trying to make a difference in rare disease?
Yeah, and I'll let Peter give his perspective on this too, because I think that you know, when we recruit people to my lab, when we have people working in my lab, it is a little bit different of a mindset, right? We are trying to push things forward. You know, our ultimate goal is not necessarily always a paper. Papers are great. They're wonderful to publish. They're wonderful to get this information out. But you know, again, one of the goals is trying to push things forward in a therapeutically relevant way. And so I joined UC Davis about 8 years ago. And as a postdoc in the lab of Jan Nolta. And Jan Nolta was recruited to the University of California, Davis. She's kind of a local Sacramento person, grew up here, went away to Southern California and St. Louis, and you know, became one of the most influential stem cell researchers in the US, and got recruited to come back to UC Davis. And really build this translational center. And where my lab is housed in the Institute for Regenerative Cures is this translational center where we have good manufacturing practice, we have a regulatory core, we have all of the pieces in place to move these things forward for clinical trials. And she recruited me from a basic science lab when I got my Ph.D. And so I really came here and started interacting. When I was a graduate student, I had never met a family affected by any of the diseases or any of the genes that we were studying. And I think I was in California for about 48 hours before I met families. And that really starts to change the way that we think about doing research. You know, it's you know, great to do things in cells. It's great to do things, but if we really can't move it forward and make an impact, or you know, hopefully, you know, do things that may change the quality of life of an individual, right? You know, then it kind of changes, right? And so we recruit people to do that. We try to develop that mindset, instill that mindset. As Peter said, we try to interact with the patients and the foundations and attend the family days if we can, to give research updates when possible. It's been a little harder with the pandemic that we haven't necessarily gotten all that, you know, face-to-face human contact that we're used to. But it's something that we're interested in getting back to doing, and you know, connecting with the families to hopefully make an impact. So I'll let Peter give his perspective on this as well. You know, kind of what drives him as a researcher in the translational world.
Yeah, I think I guess I'll start off a little bit with my background. I have always been interested in this gene therapy translational space. My background as an undergraduate, I was actually a linguistics major, and I became really interested by science. So I switched over to neurobiology. And this was actually due to sort of witnessing close family members undergo these induced seizures. So it was like a really profound experience for me about like, oh, I want to get involved with a field that could, I could make some type of impact that could help, not necessarily someone in my family, but helping others who, as a caretaker, I think oftentimes you can kind of have this feeling, oh, that's just the way things are. And I think with the advent of so many of these different technologies, that's just the way things are is not really an answer. I think there's profound things that researchers can do to push the envelope forward, to come up with things that are actionable on things that we previously thought were not actionable. So that was a big driving force for why I pursued science. I stuck on for my PhD with Dave Siegel's lab, particularly because of the interactions he had with Angelman syndrome families in the area. I very early on in my career, I was able to meet these angels, their families, and I was able to really strongly identify with a lot of the caretakers with it because, I think there's something called a caretaker's oath, which I didn't know what that was until I became more involved with the Huntington's community. But it's basically sort of do what you can to help your loved ones. And so I pursued my PhD specifically with the interest of gene therapy, translational science. What the end goal is, can you make some type of difference that can impact something? Even if like your work doesn't necessarily succeed, what can you learn from it and how can you influence others to do something better? And so coming into Kyle's lab and in the space in the Institute for Regenerative Cures, I don't think you can be, sorry, there are planes in my area, so there's a little bit of sound. But I don't think you can join our group without having sort of this drive to try to help people. And I think that's really evident in just how much we actually get to see the patients and how much they'll read about our research, they'll cold email us, what can we do, how can we get involved? And it's very profound when you see people come together to try to make a difference. And they find you and they believe in you and they believe in your ability as a researcher to try to drive things forward. And we have all these stories about people have bake sales and they'll bring like a check for like, $20 or something. And you have like, and it's really like $20 can go a long way in terms of like what we can do in lab. And they're coming and giving it to you, and it gives you a lot of perspective because yeah, I'm like driving something forward that was raised by people who care. And it gives you a ton of perspective in terms of like waking up in the day, because science can be very, very difficult. Like things don't always necessarily work out. But I think when you have sort of this perspective that what you're doing is beyond just yourself and what you can do may have impacts that can make someone else's life better in some way, gives a lot of energy to wake up in the morning to go to lab and do your thing. So that's a big driving force for why I have been in this lab for quite a while, why I stuck around. And I think that's generally the culture of what we have in the Fink Lab is people want to make a difference. And I think that's really vital to being a successful translational researcher.
Wow, that was really inspiring.
That was.
I mean, it couldn't, you say, it's the reason why we're here, because just the way it is, it doesn't have to be the answer, like my daughter Josie, when I got her prognosis, I was like, it doesn't have to be the prognosis, but then we're relying on finding you, and exactly, not just brilliant scientists, but brilliant scientists who really care. And we always say, every dollar donated, every minute spent in the lab could have an impact beyond measure. And that, we don't even know what this is going to do in, 20 years, how we're going to change the face of, the blueprint of human life for the better. So, thank you so much as parents. Thank you so much for how much you care and, being really brilliant scientists too.
And for doing this project. It's one that we're very excited about. We have spent a lot of time over the last couple of years on basic science, building our assets, getting stem cells from, skin samples from parents and turning them into stem cells and all of that. And it's for these projects. It's for these proof of concepts that if they work, we can take them, as you so nicely described, into the, from bench to bedside. So thank you for being here and on this journey with us.
We're so excited to watch this next year and see where it goes from there.
Yeah, thank you so much. I think I can speak for Colin that we're very thankful for this funding to go forward. And I've got to say, just being in the patient advocacy space, these grassroots programs are so vital in terms of funding these like ideas that it may be difficult to get funding. Funding in general is just a difficult thing to get in general, but these special interest groups that are able to provide these, whatever dollar amounts is always helpful. But coming up with an idea, having the funds and resources to go forward and develop it, and actually seeing something that's very exciting is often sort of the catalyst that's necessary to bring it forward, because now suddenly we have data to suggest something might be very effective, and now you can go towards these broader, larger funders and you can bring it forward, and it just sort of snowballs in. But that beginning step is so incredibly vital, getting it rolling.
Absolutely. And that's usually the hardest to fundraise for from institutions. And that's where the patient advocacy groups and patient dollars really make a difference.
So that's what's up with FOXG1 Research. This is such a wonderful conversation, and it's so nice just to hear it from straight from the lab. I know that all of our parents and all of our supporters are, going to be so happy to have met you both in this way. So
thank you for being here.
Thanks for being here. And, if anybody watching has any questions for Kyle or Peter, you could direct them to us. I'm, as always, Nicole at FOXG1 Research, and we will always keep you posted along the way.